Drug target interaction. See, we have seen, I have explained it to you that the drug can target the different type of macromolecules which are there in our body. Like they can target carbohydrates, they can target proteins, they can target lipids, other things. Now what you need to know is that macromolecules performs different types of functions in our body. Like we know that proteins are the ones which actually are the main building blocks of the body. They catalyzes the reaction. They in the form of certain molecular species catalyzes all the biological reactions which are taking place in our body and are called enzymes. Some of those proteins are actually helpful in the communication system of the cells. Those are called receptors. And similarly, lipids and carbohydrates we know are the main structural materials of the cell membrane. Also, they are the main sources of energy in our body. Also, we know that nucleic acids are the ones which are basically the compounds in which or in the form of which the genetic information is stored in the cells, which is being transferred from one generation to the other generation. So these are the large macromolecules which performs different functions in the body. Now, the drugs which we take targets these macromolecules. We want to study the drug target interaction in the form of the drug protein interaction or the drug enzyme interaction, because enzymes are the ones which basically are responsible for catalyzing all the biological reactions which are taking place in our body. So we have enzymes as drug targets. See, when the drug is taken, particularly keeping the target as in the enzymes, we need to understand how the enzymes works. So let us analyze what enzymes basically does. The catalytic action of enzymes. So we know that enzymes are biocatalysts. Though the enzymes are having active sites on their surfaces. What they do is they particularly attract the substrate or the reactant molecules on the surfaces, attaches themselves with those active sites, performs the reaction the reactions are performed on the surfaces, and what happens is then the substrate is broken down into two pieces or into product, and it goes off, and again it is available for the further reactions. Now, so the enzymes have certain very specific active sites. They are shape selective catalysts. They absorb the reactants on their surfaces where the reactions takes place, and then the substrates are changed into other products. This is what is the catalytic action of enzyme. Now, we have to study the drug enzyme interaction. How the drug interacts with the enzyme. Now, after understanding how the enzymes catalyzes a reaction, now let us understand the drug enzyme interaction. See, drugs can inhibit all the enzymatic activities, and the drugs which inhibits all the enzymes activity are called enzyme inhibitors. They stops the working of the enzymes. Now, how they stops the working of the enzymes, they do so by the two ways. First they can compete, the drugs which compete with the enzymes, and before being allowed or the enzymes, what I can explain it to you by this diagram. Suppose this is what is the active site we have, and this is the substrate which is actually being absorbed on this active site. A drug is provided. Now this drug, and this is the reactant or the substrate. So the drugs which competes with these molecular species, with the enzymes, and they are the ones which gets absorbed first on the active sites, and thus blocks those active sites and inhibits their enzymatic activity, are called competitive inhibitors. So those drugs which inhibits the activities of the enzyme by competing with them, and by blocking them, they are blocking their active sites, are called competitive inhibitors. Then the second way of inhibiting the enzyme activity is by allosteric sites. Now what happens is, there are certain drugs which doesn't bind themselves to the active sites. They bind themselves to some other part of the enzyme. Like suppose this is the other part we have, and this is the active site in the enzyme. Now this is the drug which is being provided. This is the drug. Now this drug binds itself to some other site of the enzyme, and when it gets binded to this enzyme, what happens is the shape of the active sites get changed, due to which it won't be able to absorb now the substrates on the surfaces, and this is how all the enzyme activity is stopped. Now such type of sites are called allosteric sites. So this is an allosteric site, and due to these allosteric sites, what happens is they can stop the enzymatic activity. So the drugs they can inhibit the enzyme activities by any one of these two. Sometimes this bond between the drug and the enzyme becomes covalent, very strong bond, and in such cases the enzyme becomes useless. They stops working permanently, and in those cases the body throws them out and the new enzymes are synthesized in those cases. So this is how the drugs enzyme interaction takes place.